All right, everybody, welcome back to class. So, have you memorized the amino acids yet? You got it? Uh-oh. Well, you better. Okay. So some of what we're going to do today is actually going to require you to have an idea of what the one-letter codes for the amino acids are. I'm not going to go through them all in detail because however you need to memorize them, it's going to involve more than me just saying the words to you. So you need to make sure you get them. I've divided them up into 4 general categories. In green, you have the non-polar ones. These mostly have hydrocarbon chains. In purple here you have the aromatic ones. These include phenylalanine with a one-letter code of F, that makes sense. Tyrosine, one-letter code Y. Why? Because we've already used T for something else. And this is like phenylalanine but has an OH group. They both are aromatic. Tyrosine has a phenol side chain, which means it's got the OH group. And its pKa is around 10. Then tryptophan is the other one. We don't worry about the side chain pKa for tryptophan because it's high enough that it's not relevant at physiological pH. Its one-letter code is W. All of them are aromatic. Now there's another that meets the criteria for aromaticity, but I included in a different list. Not because it's not aromatic, it is. Histidine is aromatic, but histidine can be positively charged at the right pH. So you've seen the imidazolium group before with a pKa of 6, and it can adopt this neutral conjugate base form, imidazole. At pH 7, it should be about 90% neutral, about 10% positively charged. So it's good to put histidine in the category of polar amino acids because it is, even though it's aromatic. So that's why I've put it there. Most of the time at pH 7, histidine you would call neutral, but there are some times where it can be positively charged in enzyme active sites and it's useful. Other than that, let's see, one other thing I wanted to draw your attention to was the concept that some of the amino acids have a branch point at the beta carbon. Valine does, isoleucine does, and then threonine does as well. Threonine has a branch point at the beta carbon. For isoleucine and threonine, those beta carbons are stereocenters, and only one stereochemical configuration is correct at that stereocenter. There are other diastereomers that are not biologically relevant. I'm not going to push you on what that correct stereochemical configuration is. Your biochemistry professors are unlikely to as well, but you should remember that there is a correct one. Let's see, one other thing to mention is that cysteine has a thiol side chain pKa of 8. The cysteine side chain is highly oxidizable. It forms disulfides with other cysteine side chains. A disulfide is where the 2 sulfurs are linked to each other. You can tell this is oxidized relative to 2 free cysteines because you're missing 2 hydrogen atoms. And oxidation in OChem can mean loss of hydrogen atoms. So this is the oxidized form of cysteine. It's the disulfide, and these are found all over the place in proteins for structural stability, especially in proteins that are secreted. Interestingly, the interior of the cell, the cytoplasm, is in a reducing environment, so there aren't any disulfides in the cytoplasm or in proteins that are inside the cell. The ER, the Golgi, and lysosomes, those are all oxidizing environments. Extracellular environment is oxidizing, so extracellular proteins or proteins that are in the ER or the Golgi tend to have a lot of disulfide bonds. And a lot of what goes on in the ER is related to formation of correct disulfide bonds. All right, so you're going to have to go with that. Now, don't worry about individual Ramachandran plots for the individual amino acids. That's mostly to facilitate our discussion. The point I wanted to make is the identity of the side chain impacts what types of dihedral angles, phi and psi angles, can be adopted by an amino acid. We illustrated that last time by looking at the impact of adding the branching methyl group on valine relative to alanine. These are heat maps, so the red color indicates lots of examples of alanine residues with dihedral angles that are in this area of the plot. This is the alpha helical region of the plot. There are also lots of, but slightly fewer examples of alanine in beta sheets. You add that beta branching group, and valine is, again, comfortable in helices, but much more comfortable than alanine in sheets. So the identity of the side chain can impact conformations adopted by that amino acid. That's also illustrated, I think I showed you glycine, which has parts of the Ramachandran plot highly populated that none of the other side chains do. And the point is that glycine lacks the side chain, and that opens up all kinds of other dihedral angle possibilities that aren't possible with the other amino acids because the side chains would bump into stuff. So understanding that glycine is highly flexible because its lack of a side chain is important. Another interesting feature is proline. This is a weird amino acid. I put glycine and proline in the weird category. Glycine because it lacks a side chain other than a hydrogen, and then proline because in proline the side chain is tied back to the amino group. Proline is the only amino acid that's a secondary amine, and when it's in a peptide or a protein, there's no hydrogen on that proline residue. The fact that you've got a ring here, remember, rotation around the bond between N and the alpha carbon is dihedral angle phi. Rotation around the alpha carbon and the bond between the alpha carbon and the carbonyl carbon is dihedral angle psi. This constraint, this ring that involves both the nitrogen and the alpha carbon, clamps down on the values of psi that the molecule can adopt. If this is the ring, you eventually get to dihedral angles that would stretch the ring to the breaking point. The ring dramatically restricts the values that can be adopted by that phi dihedral angle. And you see that when comparing the proline amino acid to any of the other amino acids. Just let's compare proline to, say, leucine or isoleucine. All of these have spots over here sort of at psi values that are getting close to 150, 160. Proline doesn't because the ring prevents it. So proline is much less flexible than the other amino acids are. Well, we could spend a lot of time talking about amino acid structure, but what I want to do now is talk about the kinds of non-covalent interactions that are available to these side chains. This is going to help us understand some things about how proteins fold, and so, to some extent, this is going to be a review of stuff that you learned in Chapter 3 of 351 when you talked about functional groups. So, non-covalent interactions in peptides and proteins. Well, one of the most obvious ones is called a salt bridge. This is an ion-ion interaction or an ion pair. It involves a positively charged side chain on one side, so we might do the lysine side chain. By the way, in the lysine side chain, if this is the alpha carbon, the positively charged ammonium group is on the epsilon carbon. So that's how you can remember how many carbons are in the lysine side chain. You got to get out to epsilon before you put on the ammonium group. That's positively charged. There are two negatively charged side chains. One is aspartate and the other is glutamate. In glutamate, let's see, I'm going to get this wrong. I always forget how many alpha, beta, gamma. Yeah, I got it wrong. Whoops. So in glutamate, the negatively charged side chain is on the delta carbon. And in aspartate, the negatively charged side chain is on the gamma carbon. So aspartate is truncated one carbon relative to glutamate. Sorry, I wrote the wrong code. This is glutamate. That's the 3-letter code. What is the 1-letter code for glutamate? E, good. I don't know why it's E, other than glutamate ends in E. What is the 1-letter code for lysine? It's K. I don't know why, but that is what it is. So the positive charge on lysine can interact with the negative charge on a glutamate side chain. You can mix and match. For example, there's other positively charged side chains like arginine. There's other negatively charged side chains like glutamate, but an ion-ion interaction where like charges attract is an example of a kind of thing you can see all the time in proteins. Okay, other questions about that? Yeah.
Well, I have a question for amino acid for proline.
Okay.
Is that the terminal, like, of a peptide chain because it cannot attach another amino acid to?
No, actually, proline is often in the middle of peptide chains. This nitrogen can be attached to other groups. You can form amides with other amino acids. The squiggly line here, this is not a methyl group that is connected to the previous amino acid. The squiggly line here is the carboxylate connected to the next amino acid.
So when is the last amino acid?
What is the last amino acid in the peptide chain? It depends on what the mRNA says the sequence should be. What is the stop? Is there an amino acid for the stop codon? No, that is an excellent question. You'll get to that in biochem. Stop codons are codons that don't have tRNAs to them. So they stop because there's nothing to fill the ribosome there. And, I don't want to say this, but I do. There's actually a technique in chemical biology of co-opting the stop codon to introduce your custom amino acid of your choice. You should look up, it's called amber suppression. You could look up the work of Pete Schultz if you want to know more about that. But anyway, okay, others? Yeah.
For the two acidic ones, like glutamic acid, could it attach at either side?
Oh, right. So the two acidic ones, glutamic acid and aspartic acid, there's a question. I'll need to draw the structure of the amino acid itself to make this a little bit clearer. Let's just do aspartic acid. This is where the acid group is on the gamma carbon. One question is, do you form the amide bond with this carboxylic acid or with this one? The answer is, if you're in the ribosome makes it go with this one. The enzyme controls that. In the lab, we use protecting groups. We protect this carboxylic acid as something else and let this one be free. How do we do that? We use the difference in pKa. So this is where pKa can be useful. The pKa of the aspartate side chain is around 4. The pKa of the backbone carboxylic acid is around 2. So there's a range of pHs where this one is negatively charged and this one is still neutral, and so you can use this as a nucleophile and protect it. We'll maybe talk about some side chain protecting groups next time. Yeah.
So do we see salt bridges as like structures that like repeat themselves? Like, is a salt bridge particularly stronger when it shows up like in proteins? Is this usually something that has to repeat to like?
So how strong is the salt bridge is a good question, and a lot of these non-covalent interactions that I'm talking about run in the range of 1 to 10 kilocalories per mole. That may not mean a lot to you unless you remember bond energies from 351, which you've probably forgotten, but a carbon-carbon bond, a typical carbon-carbon bond is worth about 80 kilocalories per mole. So these are weaker than a covalent bond. The other thing you have to do to understand whether they're strong or weak is to consider alternatives. For example, a salt bridge might be strong if the two side chains are close to each other. By Coulomb's law, the energy between those two side chains is going to be related to the distance between the two. You make them further apart, less strong. The other thing that can make a salt bridge less strong is if there are competing counterions, a lot of them around. For example, a sodium ion could pair with a glutamate, and a chloride ion could pair with the lysine, and that would tend to interfere with the salt bridge. So there's a lot of factors. It's highly context dependent, and there's more we could say here, but that's probably enough to be starting with. Others? Yeah.
So is the formation of salt bridges dependent on pKa?
Formation of salt bridge is highly dependent on pKa and pH. So you could imagine lowering pH to the point where the glutamate would be protonated, and then it's not a salt bridge anymore. There are some environments, like in your stomach, the pH is really low. In the lysosome, the pH is around 5, so close to where those glutamates might be protonated again. And that can tend to break up salt bridges, yeah. Others? I saw, yeah, no? Same, okay. Yes?
So where do salt bridges take place? Because like for hydrogen bonding, that'll help keep like alpha helices together. So like where do salt bridges take place?
Where do salt bridges take place? Generally on the surface of proteins, for reasons we'll discuss soon. Polar side chains tend to be on the exterior of proteins where they're exposed to water. Some salt bridges are buried in the interior or are in more hydrophobic environments. When that happens, they actually get quite a bit stronger because they don't have a lot of other alternatives besides interacting with each other. Protein surface salt bridges can interact with water or counter ions. So it's highly context dependent, but mostly on protein surfaces. Others? Okay, so hydrogen bonding that you just mentioned is another non-covalent interaction that happens within proteins. Classic examples of this might be within an alpha helix, in which an amide bond for one amino acid interacts with, I'm just going to use squiggly lines here, interacts with a carbonyl carbon of another amino acid at some other position in the protein. This is an example of a hydrogen bond. We'll see more of these. I'll show them. These can happen between backbone groups and other backbone groups. By backbone groups, I'm talking about carbonyl oxygen and amide hydrogen. It can also happen between side chain groups, and you can mix and match. It can happen side chain with backbone and vice versa. So as you look at those amino acid side chains, it's useful to think which ones are capable of hydrogen bonding. For example, serine, for sure. Tyrosine, yep. Tryptophan, indole NH, yep, that's capable of hydrogen bonding. Anytime you have a hydrogen bonded to an electronegative atom, that's a time where you can do hydrogen bonding. So don't be surprised. Asparagine and glutamine are neutral amino acids, but they're very capable of hydrogen bonding. The oxygen there is a hydrogen bond acceptor. The NH2 has 2 NH bonds that can participate in hydrogen bonding. That's true for both asparagine and glutamine. Okay? So there are others that we should talk about briefly. One is what I'm going to call just broadly pi interactions. To understand these, and this may be a review, I think some of you learned about it in 351 and others did not, we need to take another look at the benzene ring. Okay? This is something we haven't mentioned before. I'm just going to draw benzene, even though phenylalanine, one of these hydrogens, is going to be replaced with a carbon that's attached to something else. We're kind of looking at the benzene ring on its side, okay? One of the things you need to know is that sp2 carbons are more electronegative than sp3 carbons or than hydrogens. So that means that in benzene, the carbon-hydrogen bonds each have sort of mini dipoles with the partial negative end towards the carbon and the partial positive end towards the hydrogen. This is a symbol that chemists use to explain a dipole. The little cross indicates the positive end of the dipole. Now overall in benzene, these dipoles cancel out, and so benzene is rightly called non-polar because there is no net dipole. That's all that nonpolar means, no net dipole. However, it would be wrong to say that the distribution of electron density in benzene is completely uniform, because all of the carbons are electron rich and all of the hydrogens are electron poor. And we could maybe demonstrate that if we use the color red for electron rich. The carbons are electron rich. Let's use the color blue for electron poor. All the hydrogens are electron poor. So you have this, an aromatic ring like benzene has what we might call an electron poor edge and an electron rich face. And that kind of arrangement, it's not a dipole, it's called a quadrupole. And that gets into some vector math stuff that we don't need to worry about. But you can see interactions of various groups with the electron-rich or electron-poor portions of a benzene ring. Classic example of this is called a cation-pi interaction. So let me just demonstrate with this drawing what that might look like. Imagine you have a potassium ion. I'm drawing it and we'll color it blue because it's positively charged. It's about that size. The potassium ion can actually sit down on the face of the benzene ring, and there's a strong interaction between the electron-rich carbons of the benzene ring and that cation. That's a cation-pi interaction. And these happen a lot in acetylcholine esterases and in some other neurotransmitter receptors. They happen a lot throughout proteins. So you have to be a little careful if you're just going to look at a benzene ring like phenylalanine or tyrosine or tryptophan and basically say, okay, that's nonpolar. Because it is, mostly, but it's capable of interacting with cations because the face of those aromatic rings is electron rich. Another thing you will tend to see among aromatic side chains is what is called pi-pi stacking. This happens when the electron-rich edge of one ring contacts the electron-rich face of one ring contacts the electron-rich edge of another ring. So there's a couple of ways that you could imagine this happening. One might be what's called sort of T-shaped stacking. So here's our first benzene ring, and then I'm going to draw the other one even more sort of on its edge. And again, you're going to remember that the hydrogens of benzene are electron poor, whereas the carbons are electron rich. So it would make sense to have one benzene ring stack, the edge of one benzene ring stack against the face of the other, so that their electron-rich and electron-poor portions, respectively, can interact. So this is called sometimes T-stacking or edge-to-face. That makes sense? So you'll see that among aromatic side chains. The other thing you can see sometimes is, and I'll just draw a schematic of a benzene ring. It's a disk, and here are the hydrogens. If you remember that the face of the benzene ring is electron rich and the edge is electron poor, then this kind of offset stacking maybe would make sense. The positive, or the I switched colors. The electron poor hydrogens on one ring are over the electron rich face of the other ring, and vice versa. And this kind of thing is actually also happening in DNA. Base pair stacking in DNA takes advantage of this kind of thing. What you don't see for benzene rings is full-on face-to-face stacking, like a hamburger or a Big Mac or whatever, because the rings repel each other. The electron-rich faces would repel each other. All right. So those are some examples of the kinds of non-covalent interactions that you can find in proteins. There are others. I do some research in non-covalent interactions, so I really geek out on this. But I think we're probably close to what's useful for this introductory class. I do want to mention something else that is widely misunderstood in protein folding, and that is what we might call hydrophobic interactions. But I can't spell hydrophobic when I'm in a hurry. So I'm going to put, so if you were to look at, say, a leucine or a valine side chain, again, here's the alpha carbon, here's valine. Leucine would look like this. Alpha carbon, in leucine you've got the beta carbon and then a branch point at the gamma carbon. If you would look at that, you'd say, okay, there's no dipoles there. There's no really tendency for hydrogen bonding, at least among the side chain. Those are both non-polar side chains. What interaction do we have for non-polar groups? Think way back to 351. What have you got? Van der Waals, which means what? You're murmuring induced dipole interactions. What is an induced dipole? Well, it's a dipole that's induced. No, what that means is when two nonpolar molecules get close together, the motion of the electrons in those two molecules becomes correlated such that the electron cloud leans to one side on one molecule and the other electron cloud on the other molecule will lean in such a way as to provide a complementary interaction. So a temporary slightly positive edge on one molecule and a temporarily slightly negative edge on the other molecule close to it. And it goes back and forth, but in general they correlate the motion of their electrons to accommodate each other. One of the things about van der Waals interactions is they're very weak. So, and I don't know what the single air quote is, so they're generally very weak. So people talk about hydrophobic interactions as though they're strong. The van der Waals interactions themselves are weak. However, the hydrophobic effect is quite strong. So I can't spell hydrophobic when I'm talking. Just give me a second. What is the hydrophobic effect? You got nothing. Go ahead. Okay, so water molecules and what? Okay. Okay, that's a good way of thinking about it. All of the polar stuff wants to be next to other polar stuff. So the nonpolar stuff by default tends to get shoved together. That's a really good way of thinking about it. On a macroscopic scale, oil and water don't mix. When you're doing a recipe and you need to mix water and vegetable oil or something and you want it to not be separated, often you use an egg, which is an emulsifier and helps the two stick together for reasons we'll talk about another time. But that's the hydrophobic effect. The basic idea in the hydrophobic effect, if we could just make this really quick and simple, is imagine a protein with a bunch of nonpolar groups and they're all exposed to water. When nonpolar surface area is exposed to water, it does something interesting to the structure of water. You learned about this in 351. We don't need to say a huge amount, but what water has to do in the immediate vicinity of nonpolar groups is it sort of forms an ice-like cage around the nonpolar group. It's weird because water likes to interact with itself. It forms productive and fairly disorderly hydrogen bonds with other water molecules, but it can't interact directly or strongly with the nonpolar molecule. So a water molecule that's here, right next to the nonpolar portion, loses a hydrogen bond forming partner, and therefore it holds on more tightly to its adjacent partners. It loses a lot of entropy, it loses a lot of conformational freedom, and it's almost like each of the water molecules in the immediate vicinity of the nonpolar group form an ice-like cage. That lowers the entropy of the water molecule. If instead the protein collapses in such a way that all of the nonpolar side chains are on the inside where they're sequestered from water. Suddenly, all of those water molecules that used to be in the ice-like cage are back in the bulk water where they have the maximum number of hydrogen bonding partners and the maximum amount of freedom. And you can tell that's favorable because they are all saying, "Whee!" Now, what do we mean by favorable? Well, we mean this process of collapse so that the nonpolar side chains are all in the interior of the protein. We call that process folding, and it's driven by, as are all processes, driven by free energy, which comes from enthalpy. Enthalpy for us is going to be about interactions. Are the interactions you form stronger than the interactions you broke? Delta S is entropy, and this is going to be, is the system more disordered than it was previously? Normally, you might think the process of protein folding, of going from a random coil to something that has helical secondary structure and other things, involves a dramatic loss of disorder or a dramatic increase of order. That's true, but only if you limit yourself to thinking about what the protein is doing. But protein folding happens in water, and so you also have to consider how folding impacts water. And this is how folding impacts water. When the protein is unfolded, nonpolar side chains are exposed to the water, and water is highly ordered in the immediate vicinity of those side chains. When collapse happens, you do increase the order of the protein, but there's a compensatory and even totally erasing of that increase in order by the fact that all of the water molecules are now free. And that pays for the protein folding. Now that's a quick description of the hydrophobic effect, but the conclusion is that nonpolar side chains tend to be in the protein interior, inside a folded protein, not exposed to the solvent. Whereas the surface tends to be occupied by polar amino acids that can interact with water just fine. Questions about that? Go ahead.
So if the folding and stuff is happening largely because of these like hydrophobic effects, I don't know if it's a question inside the scope of this class, but like what does that look like when you have something that's like a transmembrane protein where you used to have these nonpolar groups on the outside of the protein so it can shut itself into a
Right.
membrane?
Yeah, so this is a question. We may get to something that's similar to this, but the question is about transmembrane proteins. So we need to draw a really fast lipid bilayer. The circles are the polar head groups for the bilayer. The tails are the nonpolar tails. What's being referred to here, ooh, I love exponential copy paste. What's being referred to here is that some proteins must cross the membrane, and they'll have an extracellular portion here that can adopt whatever, and it's around water, and then there'll be a transmembrane portion here, sometimes several passes through the membrane, and then an interior portion. Extracellular and intracellular, you're all in water, so you're worried about the normal hydrophobic effect. Not so in the interior, where the rules must change because the interior is nonpolar. And so, yeah, transmembrane segments have a different set of they will use different amino acids on their surfaces than the extracellular portions will. And it's exactly because of the hydrophobic effect. Yep. You'll learn more about that if you take biochem.
Okay, I will.
So I don't have too much more to say, but yeah. All right, so yeah. How does denaturation affect the hydrophobic interactions? Good question. So a couple of things. Temperature shows up in the, denaturation means unfolding. You can unfold in a couple of ways, actually multiple ways. One of the ways that you're most familiar with is when you fry an egg, right? So the biggest protein that's present in egg white is called albumin, and you've seen what it looks like. It's clear and translucent and goopy, and then you stick it in a pan, turn up the temperature, and what happens to egg white anyway? It goes white, right? And it goes from being sort of a sticky liquid to a solid. That's protein denaturation. You turned up the temperature, the albumin unfolded, and its properties changed. How does unfolding happen? It's kind of complicated. There is a temperature dependence to the hydrophobic effect. You can also think of it as you increase temperature, you give increased kinetic energy to the individual atoms in a protein. And if you look at any of these interactions that we've talked about, if the atoms start moving faster, they're going to be able to move out beyond a point where the interaction is worth anything. And so changes to the hydrophobic effect and to the strength of the interactions with increasing temperature really is probably what causes denaturation. I don't think I have it on this computer. I'll have to check, but maybe next time I'll show you a simulation that I did in my lab of a protein unfolding with increasing temperature, and you get a sense for what's going on. Okay, others? Okay, well, let's now talk about, with this foundation in amino acids, if you don't know them, you're going to wish you did. And these interactions, we're going to now talk about the relationship between sequence, structure, and function. And our first test case is an alpha helical coil. I've drawn for you 33 or 34 residue peptide derived, or protein, derived from a yeast transcription factor. The name of this protein, in case you're interested, or this peptide, is GCN4P1. It is peptide 1 derived from the yeast transcription factor GCN4. We're going to talk about its transcription factor function maybe towards the end, but first we need to just zoom in on this sequence. This sequence is from the dimerization domain of that yeast transcription factor. So you might expect that this sequence dimerizes, and when we're done, you're going to know why. So each of those is a one-letter code. You know what R is? If you don't, you should prepare speedily, for I would hate for you to take the exam and get caught unaware. R is arginine. That's a positively charged amino acid. So we're actually going to go through and annotate all the places that are electron poor, positively charged. The K is 2. So R and K are all positively charged. That's arginine and lysine respectively. So you can see where the pluses are in this molecule. Did I miss an R? Dad amino acid joke. What is the pirate's favorite amino acid? Arginine. Yep, good. Okay, all the negatively charged ones are E and D, glutamate and aspartate respectively. Let's see, did I miss any? I think that's it. So if we count up, and actually the C terminus of this peptide is negatively charged. The N terminus is not positively charged because it's been capped with an acetyl group. I may or may not tell you why they did that. If we count up the positive charges, we've got 1, 2, 3, 4, 5, 6, 7, 8. Negative charges 1, 2, 3, 4, 5, 6, 7, 8. Okay, so overall, at pH 7, this is neutral. Now, that might be important because this peptide is supposed to dimerize, right? Dimerization means two molecules coming together to form a non-covalent complex. If this peptide was heavily positively charged, there'd be some charge-charge repulsion in forming the dimeric complex. So that's one way that sequence here is going to influence structure and function. The other thing we need to do is highlight all of the non-polar residues, because we know that those are going to tend to want to be in the interior of a protein. So I'm going to do that just with an orange highlighter. Methionine, leucine, valine, a couple of leucines there, another leucine, another leucine, another valine, another valine, another leucine. Those are the non-polar amino acids, okay? We'll come back to this, but there is kind of a characteristic spacing between the nonpolar amino acids. This methionine is 1, 2, 3 residues or spaces away from this leucine. This leucine is 1, 2, 3, 4 residues away from the next nonpolar residue. So you have this 3, 4 pattern. And that 3, 4 pattern also shows up here. And sometimes the pattern is broken a little bit. For example, these two are right next to each other. And there's what looks like a polar amino acid and asparagine N at this point where you would normally expect, or where you might, if this were a pattern, expect a non-polar amino acid to be. But this spacing 3 or 4, every 3 or 4 amino acids, you get a non-polar one. That's really important. Before that makes any sense, though, we need to talk about a secondary structure called the alpha helix. So, and it'll become clear why we're talking about the alpha helix in just a second. Behold the alpha helix. You can see the repeating spiral as the backbone twists back on itself. And a big characteristic feature, I'm sorry, I should have explained this. This is a stick diagram of an alpha helix. The carbons are all yellow, oxygens are red, nitrogens are blue. Hydrogen atoms aren't shown because this is from an X-ray crystal structure, which on large proteins doesn't generally show you hydrogens. They're too small. But every time you see a nitrogen, that's an amide with a hydrogen there. So what we've got here is a hydrogen bond between a carbonyl oxygen and an amide nitrogen. And it's helpful to notice the interval at which that hydrogen bond occurs. You see them all down the helix. It's sort of this repeating pattern of an NH hydrogen bonding with a CO. The carbonyl oxygen, and here's how we tend to count things in helices. We'll call the carbonyl oxygen, we're going to give it the letter I. It's the ith amino acid. That's from math. Don't you hate it when they do that? Let the ith be whatever. That's the I amino acid. The next one, and remember what belongs to that I amino acid, which I'm going to color green, is nitrogen, alpha carbon, and carbonyl carbon. So the next one we would call I plus 1. The next one we would call I plus 2. The next one we would call I plus 3. And the fourth is I plus 4. Notice that this hydrogen bond that we're highlighting now in pink is between the I position carbonyl oxygen and the I plus 4 position amide nitrogen. And in general, because of that hydrogen bonding pattern, they've done the math and they've figured that there are about 3.5 to 3.6 residues or spaces per turn of the helix. Meaning that every 7 spaces or amino acids, you go through about 2 turns of the helix. Okay? And we could count, but there's 1 turn and there's another. So by the time you go through 2 turns, you basically track through 7 amino acids in sequence. Now, let's imagine what this would do for a nonpolar amino acid that is 7 spaces away from another nonpolar amino acid. Let's just go ahead and count. Let's imagine we put a nonpolar side chain here. Okay, count 1, 2, 3, 4, 5, 6, and then what you can't see here is that 7th position, right? So this would be the I plus 7 amino acid. That would be another nonpolar position there. Okay? So in an alpha helix, a nonpolar amino acid at position 1 is on the same side of the helix as another amino acid 7 spaces away. And if you put another nonpolar amino acid at the 4th position, you'd have a stepping ladder of nonpolar amino acids all on the same side of the helix, right? And that actually, if you start mapping this sequence onto a helix, what you would see is all of the nonpolar residues are along one side of the helix. It would look something like this, with all of the nonpolar amino acids along one side, and then all of the polar amino acids on the other side. Now, what do you think that helix could do to prevent the nonpolar amino acids all lined up on one face from being exposed to water? Just pair, right? Pair against the other one. Bury their nonpolar faces together. And that's exactly what this sequence does. So what I'm going to show you now is some X-ray crystallography data for this peptide. We're going to be looking first, no time for Sith eyes, but just imagine them, okay? We're going to be looking down the axis of this helix from the N terminus towards the C terminus. And here is what that looks like, okay? I will show you how this works a little bit, but for now, this is a ribbon diagram of the coil of the dimer between the two helices. I've drawn all of the nonpolar side chains as sticks. You can tell they're mostly nonpolar because they're mostly yellow, and yellow is for carbon in this drawing. And you'll notice that every turn of the helix, we come to another nonpolar amino acid. At this particular spot, a methionine residue on one helix is packing against a methionine residue on the other. One turn down, a leucine residue is packing against another leucine residue, and so on as we go down each helix. So the interface between the two helices is nonpolar, and if you look at this from the side, you have two peptides that are twisting against each other. We're going to have a little bit more to say about that sequence, but I want to talk briefly about how this relates to function. It turns out that these dimerization domains are connected to a DNA binding domain, and the two dimerization domains hold the DNA binding domains in the right shape to span the DNA duplex. That wouldn't happen if you didn't have this spacing of nonpolar amino acids. Okay, there's so much more I could tell you because I did some stuff during my PhD thesis on this and I love the subject. I probably will say more about it next time, but let's see. We need to plan ahead because I didn't quite get to where I wanted to today. I need to tell you about antimicrobial peptides and beta sheets. I also need to tell you about peptide synthesis and chemistry. We probably will need 4 days on this chapter instead of 3. So just stay tuned to the regular updates. You won't need to be done with the Chapter 23 study guide until Monday instead of Friday. All right, see you Wednesday.